Welcome to today's Explainer. Okay, so today we're diving into what is arguably one of the most high-stakes biological countdowns in human existence, and that is the engineering of a baby's first breath. When you really start digging into the data, you realize pretty quickly that the development of the human respiratory system isn't just some standard biological process. It is an absolute masterclass in microscopic engineering. We're talking about a highly precise, fiercely time-sensitive construction project where failure is literally not an option. So how does the body actually pull this off? Well, to understand it, we're going to walk through 5 major milestones: the respiratory foundation, branching out, the canalicular turning point, terminal sac and surfactant, and finally, the alveolar stage. Now, looking at this timeline, I want to point out right away that this isn't just a simple, neat sequence where one stage cleanly stops and the next one magically starts. Not at all. These stages beautifully overlap and build on top of each other. Over the next 8 to 10 minutes or so, we're going to journey from the embryonic phase on the far left straight through to the alveolar phase, which, as you'll see, surprisingly stretches out way, way past birth. And this visual right here brilliantly captures the sheer incredible scale of branching required just to make breathing possible. Look at how the body starts by forming these basic, thick conducting tubes on the left. That's our conducting zone. And then it just continuously, relentlessly branches them down into the incredibly complex, delicate transitional and respiratory zones on the right, all the way up to the moment of birth. It's essentially a massive high-speed multiplication game. All right, let's jump into stage 1, the respiratory foundation, which happens between weeks 3 and 7. So this is where the initial concrete of our biological construction site is poured. The whole entire process kicks off with something called the respiratory diverticulum. Now, to translate that dense medical jargon for a second, a diverticulum is basically just a tiny little bud or pouch. Right around the 3rd week of development, this microscopic bud of tissue just grows directly out from the wall of the foregut. And that tiny offshoot? That is the exact seed that will eventually become your entire respiratory system. Pretty wild, right? But there's a huge, immediate structural problem the body has to solve here. Because this lung bud grows right out of the foregut, which, remember, is part of your digestive tract, the body literally has to build a wall to separate the two. Looking at the progression at the bottom, you see these tracheoesophageal folds develop and then fuse together to form the tracheoesophageal septum. This wall is absolutely critical. It safely separates our trachea, our windpipe in the front, from our esophagus, the food pipe in the back. Without this highly precise separation, we simply couldn't breathe and eat safely. Okay, so once that separation is totally secure, the meticulous plumbing process begins. The primary bronchial buds form the right and left main bronchi. Then one tube splits into 2, forming the secondary buds. This gives us 3 lobar bronchi on the right and 2 on the left. Finally, those secondary buds divide yet again into roughly 10 tertiary bronchi. And just like that, the main ductwork of our lungs is officially laid out. What's really fascinating to me here is the speed at which this all happens. We are looking at a fast forward from 28 days to 56 days. In just a matter of weeks, this primitive branching network takes on the unmistakable, completely distinct shape of the human lung lobes that we all have right now. You can clearly see the superior, middle, and inferior lobes forming right before our eyes. Moving right along to stage 2, branching out. This is the pseudoglandular stage. With the foundation solidly set, our ductwork undergoes a massive expansion between weeks 6 and 16. We call this the pseudoglandular stage. Pseudo basically meaning false, and glandular referring to, you guessed it, a gland. During these specific weeks, if you were to look at the lungs under a microscope, they don't look anything like the light, airy balloons we usually imagine. Instead, they look like dense, completely solid glands. They're totally packed with thick connective tissue called mesenchyme and lined with developing columnar cells. And notice exactly why it gets this name. Look at how incredibly thick that speckled mesenchyme is around the branching airway right there in the center. During this time, the terminal bronchioles are forming, literally pushing their way through that really dense tissue. But because everything is so thick and walled off, actual gas exchange, taking in life-saving oxygen, is still absolutely impossible at this stage. Which brings us to stage 3, the canalicular turning point. Running from weeks 16 to 26, this is where the lungs dramatically pivot and start preparing for the harsh mechanical reality of life completely outside the womb. The visual evidence here is striking. On the left, you see the dense, crowded pseudoglandular tissue we just talked about. But look on the right. In the canalicular stage, the architecture completely changes. The tissue opens way up into these wide, tube-like structures called canaliculi. And notice the intense invasion of those tiny, dark red spots. Those are blood capillaries aggressively flooding into the newly thinning tissue. So the stakes are incredibly high right now. The respiratory bronchioles are elongating. The cuboidal cells that line these airways begin to flatten out, basically trying to get as unbelievably thin as possible. Combine that with the intense rush of capillaries we just looked at, and the lungs successfully construct the very first primitive blood-air barrier. Because of this specific architectural marvel, by the end of week 26, the lungs can actually support respiration, at least to a limited extent.
Now for stage 4, the saccular stage, focusing heavily on terminal sacs and surfactant.
Look, limited respiration isn't going to cut it for long-term survival, which brings us to the absolutely crucial interior finishing phase from weeks 24 to 38. Success here hinges entirely on one specific chemical, surfactant. While its production quietly kicks off in the previous stage, it absolutely must hit its peak during this saccular stage. Surfactant is essentially a fluid that coats the inside of the newly forming air sacs. Without it, the surface tension would just be way too high. Those tiny new air sacs would stick together and completely collapse upon that very first breath. Think of it as the biological equivalent of an anti-stick coating for your lungs. It is a total game changer. And notice how perfectly engineered these terminal sacs become? The blue outline is the incredibly thin squamous epithelium. Those red shapes? Those are the blood capillaries. And look at how those capillaries are literally bulging right into the walls of the terminal sacs. The body is effectively forcing the blood as physically close to the open air space as possible to guarantee maximum, highly efficient gas exchange. Down at a cellular level, the cells themselves are specializing for the heavy lifting ahead. The primitive epithelium differentiates into type 1 and type 2 pneumocytes. Type 1 cells become that ultra-thin structure we need for gas exchange, while type 2 cells act as the tiny factories churning out that life-saving surfactant we just talked about. The primitive alveoli are now perfectly equipped to keep a baby alive. Finally, we reach stage 5, the alveolar stage, where we master breathing.
So this final stage actually holds a pretty surprising twist about human development. To maximize oxygen intake, the lungs have to drastically increase their surface area. And they do this by growing secondary septa, which are essentially tiny biological dividing walls that grow right inside the primitive saccules. It's kind of like taking a giant empty room and putting up walls to divide it into a bunch of smaller ones. This subdivision transforms primitive sacs into mature, highly vascularized alveoli, exponentially increasing the amount of space available for oxygen to jump into the bloodstream.
And to put the scale of this division into perspective, prepare to have your mind blown. 300 million to 500 million. Yeah, that is the mathematical scale of mature alveoli the body must actively produce to reach full functional respiratory maturity. It's just an astronomical feat of cellular multiplication.
But here is the truly fascinating part. Actually, scratch the idea that all of this finishes up in the womb. The alveolar stage begins at 36 weeks of gestation, sure, but it continues relentlessly until a child is roughly 8 years old. So, the next time an 8-year-old complains that they're tired, well, they actually have a pretty solid biological excuse. They are literally busy finishing the construction of this invisible biological machine years and years after their first breath.
Okay, so let's quickly recap the incredible journey we've just taken. We start with the embryonic foundation, forming our primary tubes. Then we branch out densely during the pseudoglandular stage. We hit that vital turning point of vascularization in the canalicular stage. Next, we prep the terminal sacs with our anti-stick surfactant in the saccular stage. And finally, we multiply our surface area into the hundreds of millions during the prolonged alveolar stage. It is, from start to finish, a perfectly sequenced biological countdown. I really want to leave you with this one thought. Knowing the sheer architectural precision required just to keep you breathing, and knowing that your own lungs spent the first 8 years of your life quietly building up to 500 million tiny life-sustaining air sacs, how does that change your perspective on the resilience, the ongoing hidden miracles, and the deep, deep complexity of your own body? Thank you so much for learning with me today, and keep exploring the incredible engineering happening right inside you.